So let's start with the T cell receptors and with B cell receptors library preparation. And first of all, before we dive into the library preparation and all the techniques that can be used there, I would like to discuss a little bit with you what is a T cell receptor and what is the B cell receptor, what is the principle. So let's recapitulate a little bit the basics of the immunology. As you may know, the lymphocyte compartment can contain the well-known T cells. And the T cells, they have on their surface specific proteins that are called T cell receptors. So one T cell would have only one type of the T cell receptor, and the T cell receptor allows this T cell to recognize its antigen. As you may know, T cells are largely involved into the adaptive immune response. That's why the TCR is the structure that will have this T cell to recognize the peptides, the antigens. If we take a look on the TCR, on the T cell receptors, looks like this. It has an antigen binding site that will bind the antigen. And this part of the TCR would be unique by each T cell because each T cell would have only one antigen to bind. The TCR cannot bind antigen that is just soluted in the solution, just diluted in the solution. The TCR must have this antigen presented. That's why the second part of the binding is the so-called major histocompatibility complex, or MHC1 or MHC2. This is the molecule on the surface of the other cell that will present the antigen here to the T cell receptor. So the take-home message from this part is that a T cell has a TCR, and TCR helps this T cell to recognize the antigen. It's like basic immunology that you remember during the talk today. So when we come to the next part of this slide, to the B cell, it's pretty similar. You may see here schematically a B cell that is carrying its own B cell receptors, surface molecules that will help this B cell to recognize an antigen. And as you may see, this B cell receptor looks really very similar as an antibody. And in that case, it is a monomeric antibody that is just built into the membrane of this B cell. So the B cell receptor has also an antigen binding site that would be also unique for each B cell receptor within the individual. And the only difference to the T cell receptor in this case is that the B cell receptor can bind an antigen that is just diluted that is just floating in the solution. It means B cell receptor doesn't need to be this antigen presented by the other molecules as here in the TCR. So let's just recapitulate and summarize what we talked about on this slide. The T cells have TCRs and they enable them to recognize an antigen. B cells have BCRs and they enable them to recognize their antigen. So as you can see, these TCRs and BCRs are molecules, are proteins that are located on the surface of the B or T cells. And these molecules, these proteins, in order to be synthesized, that should be coded somehow, yes? And as we know, this information is stored in the DNA. DNA will be transcribed into the messenger RNA, and messenger RNA will be translated into the TCR or BCR. So as you can see, the principle is very similar. We can target specifically the messenger RNA for the TCR or for the BCR, and that's how we will gain the information about the TCR that is residing in the T cell or about the BCR that is residing in the B cell. So let's take a look. How do we prepare this library? How do we prepare the TCRs and BCRs library? First of all, let's talk once more about the single-cell RNA sequencing with the help of the next protocol. We talked about it very extensively on the day one, so let's repeat it once more. Let's imagine you have your sample, like blood sample. In that case, it could be PBMC or T cell isolates or B cell isolates. It could be also some suspension out of tissue. And the next thing that you would be doing is just loading it onto the 10x Chromium controller. So this machine enables us to generate, with the help of microfluidics principle, the single cell droplets. And in each of the droplets, you would have enzymes, you would have beads, microparticles, and of course, cell. Afterwards, when you generate your single cell droplets, you may do the reverse transcription PCR of the messenger RNA, because that is what we are targeting in the at the RNA sequencing method. What you are doing, you are binding your messenger RNA molecules and you convert them into the DNA molecules with the help of the reverse transcription. Afterwards, you amplify your complementary cDNA that was produced by this reaction, and with the help of the PCR, and you get your first cDNA library. So that is something that we heard like maybe 10 times on the day one, and what we could repeat once more today. So what is the principle by the TCR and BCR library preparation? Basically, we are doing something very similar. In parallel to the synthesizing of the gene expression library, we may synthesize a library that contains information about TCR and BCR exclusively. So how can we do it? When we build the primers, that's specific primers that may target only the sequences of the messenger RNA that was converted into the cDNA that are coding only for the TCRs and BCRs, you may enrich your cDNA by the help of these primers to get only the information about the T cell and B cell receptors. So I will repeat once more, but we are doing nothing more as just enrichment with the help of the primers of the TCR and BCR coding cDNA, out of the cDNA that we synthesized before here. So in this way, you're creating a second library that contains information from the cDNA about the TCR and BCRs. And at the end, when you fragment and purify your library, you will get two outputs. One, just normal conventional RNA gene expression library. And the second one would be the library that contains information about T cell receptors and B cell receptors. So this is the same messenger RNA related principle that just comes on top by enriching the cDNA that is coding the TCRs and BCRs. So just a little modification, but this little modification can give you a lot of information in terms of immunology. Afterwards, you sequence your information and you can start with the bioinformatics analysis. So the next question, of course, is why should we construct BCRs and TCRs? Why should you target these molecules on the surface of the T and B cells? So let's take a look at the basics immunology. When a T cell gets activated and recruited into the inflammation and overcomes its immune response, it can also be clonally expanded. So that means out of one naive T cell, we are getting a lot of T cells that are all clonally related. That means all of these T cells will have the same T cell receptor. This is called clonal expansion of T cells. The same you can observe by the B cell compartment. When the B cell gets activated and will be recruited into the inflammation, it just expands, it will be clonally replicated, and you will get a lot of B cells that will carry the same B cell receptor. So as you can see, this is something that can help us to track this expansion. When you have the information of your BCRs or of your TCRs, you can track how many cells you would have within one clonotype, how many cells will carry the same TCR, and that's why you can estimate on the clonal expansion of these cells. And when you take a look into the data, you will really be very surprised that this clonal expansion information correlates really very well with the state, with the functional state of the T or B cell. You will see that the ones that are the most expanded will be the most aggressive, the most inflamed orientated, and you will see just a real gradient out from the naive T cells that are not expanded to the inflammatory ones that are the most expanded. So this is a nice thing to have, of course, because on that way you can measure even the activity level of your T or B cells by the expansion of these clonotypes within the cluster. The next thing that is really very useful in the immunology is that with the help of the BCRs and TCRs, you can even track the T cells or B cells with the same receptor between different compartments. For example, blood or tissue. What do I mean with that? You can do two separate experiments. For example, you collect blood of the patient and you can also collect cerebrospinal fluid of the patient or some tissue suspension of the patient. And these two compartments will, of course, have two different type of cells. Maybe they will have different composition of cells, but the very specific part of the immune cells is that they can be located in both of the compartments. And if you track the same cell, for example, in blood, and then you find the same cell in the tissue, it gives you a lot of data that can be used on top of your analysis. Yeah, it can give you a lot of the suggestions. Okay, maybe this T cell was migrating into the tissue. So it gives you the new approach to identify maybe some markers that can help this migration. Or the same by the B cells. Yeah, it can also, you can, for example, make an analysis of the cells that you found in the other compartment versus the cells that you didn't find in the compartment, and so on. So as you can see, it's a very useful tool to track the same cells from the same clonotype because they will have the same BCR or the same TCR. The other cool thing is about this is that you can track the same T or B cells from the same clonotype, even between different experiments. For example, if you have a cohort of patients that will be treated, and then you can make a sample from out of blood of this patient before the treatment and after the treatment. And with the help of these BCRs and TCRs, you can track the same clonotype and then maybe measure that some of the cells will just decrease in their activity and the other ones will increase and so on. So it's a very useful tool to even compare the samples between different time points. And the next one that may be happening in the future is the sequence of the T cell receptor and the B cell receptor may possibly lead us to the antigen that it is binding. As you may know, proteomics is really very complicated as hell, and to model the appearance of this TCR and BCR, it takes just a really great effort for the computational scientists. But with all development of such technologies like AlphaFold and so on, I think it will be in the future possible to predict the antigen that this TCR or BCR may bind just out of the sequence of the TCR and BCR and other helping molecules. So now it is not so easy. It's very uncertain with the antigen prediction out of TCR and BCR information. It's not so exact. You may do it, you can do it, you can compare with the public databases, but still it's not something that can give you 100% certainty. But I think that in the future, we may come to the point where we can predict the antigen that is bounded by the TCR or BCR just with the sequence of these molecules. And in case of TCR, of course, you need to involve the MHC1 or 2 as a binding partner. So you can see a very simple method that can give you a lot of new insights in the immunology and can give you a lot of new possibilities to track your cells, to make your data more differentiated, to make a possibility to put even more questions in your scientific research. So just keep in mind, this is something that you really need to do when you're working with immunological B and T cells. It's a very helpful tool, and just keep in mind it's really it's something very useful. Additional costs in this case would be, I may say, maybe like 10 or 15% that you will need to pay more in comparison to the RNA sequencing technologies, but it's really worth it. It's really worth it because of the possibilities it can give you. So now we're finished with the TCR and BCR sequencing, about this on-top technology to the conventional single-cell RNA sequencing that you can do simultaneously with the single-cell RNA sequencing. Now we are coming to the next technology that allows you also a very good insight into some new features that may be not viewable with the messenger RNA. And now we come to the cell surface proteins library preparation. So we will talk about the second method that allows us an approach towards multi-omics, towards different sources of our information. So let's recapitulate, let's repeat the basics of the molecular biology. As you may know, DNA can be transcripted with the help of the RNA polymerases to the pre-mRNA and then into the messenger RNA. The messenger RNA can be translated to the polypeptide sequence and in the end to the protein. So you have this central dogma of the molecular biology that the way of synthesizing proteins goes out of the DNA, then through the messenger RNA, and then at the end to the protein on itself. And of course, as we talked about it on the day one, you may know that the single-cell RNA sequencing techniques that allows us to produce large amounts of information about the genes that are expressed in the cell, they are targeting, especially here, this part of this central dogma. Yeah, that means the single-cell RNA sequencing techniques are targeting the messenger RNA molecules. So in that case, when you're targeting only the messenger RNA molecules, that means that you are getting only an approximation to the actual proteomic information in the cell. Because there might be some situations when the messenger RNA molecule is present at this certain point of time, but due to some interference mechanisms or other stuff, this messenger RNA molecule will just not find the way to the protein. Yeah, there might be some situation when the messenger RNA molecule is there, but the protein will be not synthesized. On the other hand, there may be some situation when you have proteins already synthesized, but you don't have any messenger RNA molecules that can correspond to this protein, because the protein is already synthesized and the messenger RNA is not there at this exact point of time that you are doing your experiment. So this discrepancy between messenger RNA molecules and proteins can really play a very bad play with you. For example, when we are talking about the RNAseq-based method, as you understood, we are talking about the information that is based on the messenger RNA, and it's only an approximation to the actual proteome. And it becomes very apparent when you are working with immune cells, because a lot of the receptors or proteins that are expressed on the surface of these cells and can define their cell type may be already synthesized, and you will not find any messenger RNA molecule that can correspond to this protein on the surface of the cell. So, and that's a problem. For example, in our research, we saw that sometimes when we are analyzing CD4 T cell subsets, here you see a lot of clusters, 6 clusters of CD4 T cells that we were analyzing in our data set. And here, what you can see is a violin plot. That means in this plot, the dots that we can see is the level of the expression in each single cell that is plotted just on the logarithmic scale. And you may see, for example, here in the cluster 2, 3, 4, and 5, 6, the level of the CD4 messenger RNA is very high, and you can see a certain expression of this messenger RNA in these 5 clusters of cells. But one cluster of CD4s doesn't express any CD4 messenger RNA molecules at all. And when you take a look at the actual CD4 protein level, when you look into the proteomics, you may see that, wow, CD4 first cluster pops out. Yeah. So what I want to tell you with this, there are some situations where the protein is there, but the messenger RNA molecule not. And that's a problem for us, for our research, especially in immunology, because it's quite an often that you will have such a situation in that case. So how can we access this information? How can we simultaneously access not only the messenger RNA information, but also the protein information of each certain cell, and do it really with high throughput automatically and simultaneously to our single cell RNA sequencing? So there were some wise people that asked this question themselves, and that's why 2017 there emerged a publication in Nature Methods from the New York Genome Centrum. And what they made is basically a method, a principle that allows you to gain information simultaneously from the messenger RNA that is contained in the cell, but also about the cell surface proteins that are expressed by these cells. So basically you have two parts in that case. You have a transcriptome and also the proteome information. The method was called Siteseek, and Siteseek stands for cellular indexing of transcriptomes and epitopes by sequencing. So basically you have this messenger RNA as transcriptome that is being targeted by this method, and also the proteins, the epitopes, by the help of this sequencing. A very nice technique that I would like to talk to you about a little bit more with you. This technique, this principle was adapted by many other techniques afterwards, and what I will be talking with you about is a commercially available technique that is based on this Siteseek principle, and it's compatible with the 10x single-cell RNA sequencing protocol. So as I said, we will concentrate ourselves only on that method that you might use in your lab, and I may think that it's maybe better that we just concentrate ourselves on some commercially available methods. And that's why let's talk about one method that will allow you to gain the information from the transcriptome and also from the proteome of each cell. So how do we prepare this library? How do we prepare this epitope cell surface protein library? How do we access this information? First of all, you need, of course, to prepare your sample. For example, blood sample or cerebral spinal fluid sample or some tissue suspension, the suspension of cells out of tissue that were dissociated. And on the next step that you will need to do is to stain your cells in the suspension. You will be staining the cells with the specific antibodies, as you may see here on the picture. The commercially available antibody that is also that can be combined with 10x protocol is the antibody of the BioLegend. It's called Total-seq TM antibody. And this antibody is, of course, targeting some of the cell surface proteins on this cell. It may be, for example, CD3 or CD4 or CD8 and so on. There are a lot of antibodies already available on the market. And this antibody will not handle some fluorochrome or some enzyme as it normally in immunofluorescence or immunohistochemistry. This antibody will have something else. This antibody will have here an oligonucleotide sequence that is binding to it. So as you may see, this oligonucleotide sequence is very similar to the ones that we saw in the beads. So you will have a PCR handle in this sequence. You will have a specific antibody barcode in this oligonucleotide sequence, and also the poly-A tail. So sounds really a little bit familiar because poly-A is something that is also presented by the messenger RNA molecules. So that means that this poly-A tail may be also targeted by the beads with the help of the poly-T sequence. So basically what you will have an antibody that is carrying the oligonucleotide that can be targeted in the next step with the help of the reverse transcriptase reaction and with the help of the beads. So in the next step, you will load your cells in the 10x machine, build the single droplet suspension out of your sample, and then all of the beads will bind the messenger RNA molecules, but also these oligonucleotide sequences of your antibodies. So when the oligonucleotide sequences are binded by the beads, the next step that you can do is the reverse transcription. It means you will convert this oligonucleotide sequence into the cDNA, and simultaneously we'll do the same with the messenger RNA molecules. So what you are targeting with this are two types of the information: the transcriptome of with the help of the messenger RNA targeting, and also the proteome with the help of the targeting of these nucleotides, these oligonucleotides that are binded to the antibody. So as I think you see the principle, and you see maybe the point that I'm leading to you. So namely, after we can fragment and purify this cDNAs library that we've got from the reaction, we are getting two libraries at the end. The one that contains the transcriptomic information, the gene expression library, and the one that is containing the information of these binded out antibodies. So that means the cell surface proteins library, or the proteomic part on that case. A very cool technique that allows you this multi-omic approach and to fill the gap between the messenger RNA molecule and between the protein molecule that is expressed by the cell. So what are the possible application fields of this technique? Of course, the most obvious one is to label the cell surface proteins on itself. For example, you would have a cell, like in this case, it would be CD4 T cell with the proteins like CD3, CD4, also some other proteins like CCR7, etc., etc., etc. And you have a specific antibody cocktail against the cell surface proteins. So you bind them with the help of these proteins, and then you get, by the sequencing, the information about the proteins that were bound by the antibodies. Very cool technique, and can be used, this information can be used afterwards to better separate and specify your cell types, your cell populations in the sample, and it may be very helpful, especially in the immunology. Because, as I said, it's really often the case that you will have the protein synthesized, but no messenger RNA molecule that is corresponding to it. So you need to reach out into the both types of the information, into the transcriptomic, but also into the proteomic information on that case. So the next possible application field is not so obvious, but might be helpful for many of you that will be working in the single cell transcriptomics field. With the help of these antibodies, you can demultiplex your combined samples. So let's imagine the situation that you wanna increase the power of your experiment. You wanna do everything statistically significant. So that means that you would have maybe an biological triplicate. Yeah, that you will have sample 1, sample 2, and sample 3, and all of them are in one condition, in the same condition. And as we may know, especially the 10x-based techniques, they are pretty expensive. They are very cost. They carry very high costs. So it might be really expensive for you to do 3 separate experiments for each of the sample that you will have in this triplicate. What you might do is the following. You can take the antibodies, the special hashtags antibodies that will bind your sample cells. For example, here in sample 1, you will bind everything with the hashtag antibody 1. In the sample 2, we'll bind with the hashtag antibody 2, and the same in the 3 with the hashtag antibody 3. And these hashtag antibodies will be stained separately in each of the samples, so that means you have no cross-contamination between the samples with these antibodies. They will bind unspecifically, they will just bind the cells in order to mark them. And in the next step, you can pull all your 3 samples as if it would be only 1 sample and then load it onto the chip. So that means instead of the 3 different experiments, you are doing 1 with the combined 3 samples that were labeled with these antibodies. In the subsequent bioinformatics analysis, you can do the following. You can demultiplex your sample cells according to the sample and according to the expression of this antibody. This is possible. That works really very fine. So at the end, you would have, with 1 experiment, the whole triplicate of your samples that were labeled by these antibodies, and the information of these antibodies will help you to trace the origin of this cell, whether it belongs to sample 1, to sample 2, or to sample 3. Very nice techniques that spares you a lot of money, and that helps you to plan your experiments even better. So the next question that I want to address is there only a possibility to target only the cell surface proteins that are expressed on the plasma membrane? And recently there was a publication, 2021, where the expression of the proteins that were located in the nuclei were targeted. So that means in the near future, we will experience also a development in this field that you might not only target the cell surface proteins, but also the proteins that are located within the cell. It's complicated. It's more complicated as to target the cell surface proteins, but it is possible. For example, here in this paper, they described a method to target the intranuclear proteins, and with the help of this method, they revealed some new transcriptional factors that may play a role by the regulation of the mechanisms in their targeted disease. So, as I said, in the near future, I'm really certain that we will have a lot more techniques allowing us also to target the intracellular proteins and to gain even more proteomic information out of the cell, simultaneously to the transcriptomic information of these cells. So let's take a look at the end of this Siteseek part to the problems and also to the benefits of these techniques. So let's start with the negative ones. And the first problem that you may see is, of course, that you need to know the protein that you want to target. It's not an unbiased approach. It's not like a transcriptomics where you are targeting everything. You need to know the protein that you want to label. You need to know that you're working, in the case, if you're working with the T cells, that you will have some CD4 T cells in there, CD8 T cells, and you need to know the target that you want to bind with this antibody. But there are some solutions now. For example, the BioLegend is given a possibility to buy the ready-to-use antibody panel mixes that contain 137 different antibodies that you can use at once. And they are ready to use. They are equilibrated for the PBMCs. And in that case, you will have a huge panel of 137 antibodies that might give you, of course, more information about the proteins that are expressed. So I think in the future, we will have even bigger solutions with these antibodies because, as we will see in the benefits, that you can use a lot of antibodies simultaneously. The next problem is that because of the staining part, you need, of course, to adapt your protocol in correspondence to your tissue that you're working with. It's not so easy sometimes. Sometimes from our experiments, some antibodies just don't work. It's really hard play with this, with the whole concentration of the antibody that you are using, and you cannot stain too long. You cannot do a lot of manipulations with your cells. So that's why it's not so easy sometimes to adapt the protocol to your specific cells. But if you're working with PBMCs, it's a jackpot, because PBMCs, the protocol, all the protocols that are coming with the antibodies and concentration, they are based on the PBMCs. So in that case, it's not a problem. But if you're working with some exclusive tissue or some exclusive compartment, it's a little bit hard at the beginning to adapt all the protocols. And the third one, of course, and the most crucial one, it's adding a lot of the sequencing cost on top that you will be paying, because when you will take a look at the overall cost, it takes like 25% of overall costs for the sequencing. It might be helpful and worth it if you are combining a lot of samples into the one sample that you will proceed, like we were talking about in the previous slides. On the other hand, if you're doing it separately for each and every sample that you are processing, and in the case when you are like having 137 antibodies, it's a lot to pay for the sequencing that may come additionally to all the costs. The last but not least is that these antibodies, as each antibody would have some background signaling. And what I'm showing you is also a Weyling plot of the expression of these antibodies. But in this case, I plotted in our data the CD56 antibody. CD56, as you may know, is the marker for the natural killer-like cells or natural killer cells. And in that case, you see here a clear population killers that is expressing this antibody on high level. And it's cool, it's nice, you can see this enrichment here. But in all the other clusters that are not killer-like or doesn't belong to the killer population, you may see also some background signaling. So this is the next problem that you will need to deal with, where to set the cutoff. Where can you say that your antibody is positive for a certain cell population, and where can you say that it's a background signaling? Something to play with. It helps a lot if you're doing also the isotype control in parallel to it. It helps a lot if you are, for example, comparing two antibody signals simultaneously. For example, we are doing it for T cells, where you can compare simultaneously a lot of the antibodies in their signal, and then work either with the relative values between cell clusters on itself and cells in itself. So that were the problems of these techniques. The benefits, of course, are really very nice. And the most cool benefit is, of course, that the number of possible antibodies is just theoretically limited by the number of possible barcodes. And if you remember the picture with this antibody and oligonucleotide sequence, you may remember that this oligonucleotide sequence is quite long. So that means the number of possible antibodies that you can simultaneously use is in millions, even if not more. So that it's a large benefit, for example, in comparison to some fluorochrome-based techniques where you need to watch either you get an overlap in the signal of the fluorochrome or not. In that case, you won't have something like this, because the only signal that you are measuring in each of the antibody would be the barcode, would be the oligonucleotide sequence. And this sequence varies a lot between the antibodies, allowing you to use a lot of the antibodies simultaneously without disrupting or majorly disrupting the information gained by each and every different antibody. So, of course, this information brings you a new source. You are getting your information not only from the transcriptome, but in parallel also from the proteome. So it's just pushing you towards multi-source approach, which is really good when you remember the gaps between the messenger RNA and the protein information. And when you're using simultaneously the information from transcriptome and also from the proteome, you might see that the separation of your cell clusters will be drastically improved. You will see that you might detect some cell population that you even didn't think about. And especially with the new version of the CRAT, you might cluster your cells based on the information of the transcriptome and also based on the information of the proteome together. So it is pushing us towards multi-omics principle, as shown in this picture, for example. As you might see, some T cells may express CD3 a little bit lower on the messenger RNA level, but if you look into the proteomic, into this antibody level, you might see that the expression is much, much stronger and higher. The same, for example, with the basic marker for the B cells. CD19 is the marker that can allow you to identify some naive B cells, and you can see that the messenger RNA signal is quite low here. But on the other hand, if you're using the proteomic information, you will be experiencing this increase of the signal with the help of the proteins. And as I said, CRAD version 4 now enables to simultaneously use the transcriptome, but also the proteome information together to cluster your cells, to build your cell, to separate your cell populations from each other, which is really cool and which is really helpful for you, especially in the immunology, to see better the differences between the cells that you have and to reveal into novel cell populations that you even didn't think about. So on this point, I would like to thank you for your attention. I would like to make also an ending of the theoretical part of this webinar regarding the TCR and BCR-seq and also about the Siteseek. I hope that you liked this part and you gained a lot of new information in that case. And that's why I think it would be also nice to handle to the Q&A session, because we have like 10 more minutes before the Julia will start with the second part of our tutorial today. So thank you very much once more. And then I think it's time to read the questions.
Yeah, thank you, Vladislav, for such lovely introduction to Siteseek and related methods. So we already have some questions in our chats. So just to make the clarity, to provide the clarity, participants are asking, actually, like you highlighted it in your talk, but how does the protein expression exist without mRNA? I suppose it's just the question of understanding, because yeah, it's just about the way of thinking about it. Could you please elaborate on this issue? Yeah, like, Dmitro, help us with technical stuff. Moment. While Vladislav is connecting. So I will just add from my side that actually we are not saying that there is no, direct translation from DNA. We are saying that if you're looking into cell data set and and able to see, to look at this mRNA and both proteins, it doesn't mean that it's directly correlated. So actually what Vladislav said is that there is no correlation between mRNA and proteins. Direct correlation in most of cases. However, for some of molecules, this correlation in particular cell types is pretty strong. So as soon as we will solve our technical issues, we'll be able to unmute Vladislav. Okay, Vladislav, what's going on?
So now I'm unmuted.
Yes.
Sorry, I just muted myself because I was drinking, and I don't like when people hear when I'm drinking, and I forgot that I cannot unmute myself afterward. So I can just support that you said that there are certain, or what we are talking about in the case of the transcriptome information is, of course, a moment that we are taking this messenger RNA and we are targeting this messenger RNA. So it doesn't decline that in the past, like maybe few hours ago, this messenger RNA molecule was there, the protein was synthesized, and afterwards the messenger RNA molecule just was degraded because you don't need it more when you have the protein on itself. And the protein can, of course, be there much, much longer as a messenger molecule on itself. So that's why you can, when you're talking about the transcriptomics, it's just in German, the people say Momentaufnahme. So that means it's just a snapshot of this moment. And there are some cases where the protein is there and already synthesized, and it's expressed by the cell, but the messenger RNA molecule was just degraded because the protein is already there and you have a negative feedback loop in that case.
Yeah. So I see that our list of questions increase intensively. So I just would like to comment on some technical part. So one of the last questions was about what is the basis of clusterization of the graphs in the presentation. So actually these issues will be covered on my talk, and probably you'll really feel this taste of doing clustering of data and so on and so forth on the workshop. So that is why it's not covered. It's like two particular questions here. Of course, very important, but still, it was not the main goal of today's presentation, today's session at all. So yeah, and so that's all of these technical questions, and I will ask the last question from the chat just because I want us to have a break as well. So the question that I like is that, is it possible to use for primer matching part of mRNA encoding a conservative region of antibody?
Can you repeat it once more? Sorry, my internet lagged a bit.
Yeah. Like, so it's from our chat. So is it possible to use for primer matching part of mRNA encoding conservative region of antibody?
The conservative region of antibody, but of which antibody that you are using for the Siteseek in that case, or that are we talking in that case about BCR sequencing, or are we talking about the Siteseek?
Yeah, I also think that we should continue the communication with author of this question on this Slack.
Because
so
it's a bit to specify this question a bit. So maybe you can, as I said also before, you can write all your questions in Slack. And as in day one, I will be also reading all the questions and try to answer them afterwards. So we'll get all your questions also answered in the Slack after.
And I will merge this question, as we did yesterday, into one message where each of us will be able to answer that question and continue the talk. However, one small question from participants. Is there a...
Sorry, may I answer one question, because I saw this one, it's referring to the presentation on itself.
Sure.
So maybe it's just a very short question, because somebody asked whether these samples can be from different tissues or not. Yeah, of course. You can combine any samples that you want. You can combine, for example, samples from the same tissue, and then do it as in order to gain your triplicates, your biological triplicates. Or we have also done in our lab combining the sample from the lymph node, from the spleen, and also from the brain of mice, and it worked as well. So the only thing that you just need is just a proper cell suspension afterwards and demultiplexing of these hashtag antibodies.
Yeah. So, and the last question about the samples as well. So is there any limit of the number of samples that can be simultaneously analyzed?
You mean by this technique with the demultiplexing, as I understand?
It was like a general question. I mean, I'm not sure as well. So is it related to TCR, BCR, or Siteseek? So if you could explain a little bit more.
I mean, I think it's related to this sample demultiplexing, because it's the most hot part also in that case. Yes, there is a limit, and this limit is dictated by the number of cells that you can load on chip. Because afterwards, all your samples will be pulled together and then loaded in one well in this 10x controller chip and will be sent to the microfluidics. And 10x is saying that you, in order to have not so much doublets in your sample, you might load maximally 20,000 cells onto the chip. So basically, if you have your samples triplicates, you would load like 6,000 cells per each sample in that case. But the numbers of samples is just limited by the total number of cells that you can load into the 10x controller. You can even take 5 samples, but in that case, you might consider that you sort only 4,000 cells per sample in that case. So you just don't over-exceed this 20,000 mark of the maximal cell load that you can load on the chip.
Yeah. So I think we can finish our session 1 for today and have a small 5-7 minutes break. Thank you so much again for such lovely introduction to Siteseek, and I hope that now for our participants and for me as well, it was very informative, and now we are much more skilled and knowledge-rich in this direction. So let's have a break, and then after the break, we will start with the second lecture from Yulia Sitnikova. So see you in 5 minutes.
See you. Thank you very much.
Thank you a lot.
Okay, hello everybody. My name is Yulia, and today I will be talking about epigenetics and the ways to profile epigenetic marks and epigenetic states with single cell approaches. So I would like to thank before my video to organizers of this call, and I think it's a wonderful initiative, and I'm proud to be able to present there. So just a moment, my slides. Why we're actually talking about epigenetics? As you know, DNA information is more or less the same in most cells, beyond immunology, in most cells of the organisms. However, the realization of this information differs a lot. And I will remind you the way of realization of gene expression. So everything starts with transcription of RNA on the template of DNA. And then RNA, after splicing and processing, is serving as a matrix for protein expression. And this variety of different proteins is serving the base of a variety of cellular phenotype across the body. So gene expression, RNA expression is actually important for proper cell differentiation and as well during disease development. So there are a number of mechanisms which rule gene expression regulation, and this altogether they are named epigenetic mechanisms. So this is a big variety of different pathways, starting with compactization and modification of chromatin and chromatin units, nucleosomes, modification of DNA, different non-coding RNAs, and also post-transcriptional regulation, namely length of poly-A, variety of small RNAs which rule post-transcriptional gene regulation, and so on. So all this are basically united by term epigenetic mechanisms. And in today's lecture, I will focus on some most popular ones, which are most often used for single cell profiling. So the idea is that by understanding the epigenetic mechanisms and the difference in the cell population, we could figure out the gene expression networks across gene population, and actually how genetic information is realized. I'm sorry. So I will stop on few epigenetic mechanisms. First of all, chromatin compactization. So you probably know from school and university terms heterochromatin and euchromatin. Heterochromatin is highly compacted and not active. Euchromatin it has a small number of nucleosomes, are not compacted and more active. So basically, principle is very easy. As more DNA is compacted, as more difficult for it to be expressed, as less active transcriptionally it is. So therefore, scientists were developing already since long different ways to assess chromatin compactization in the cells. And it started even before the era of next generation sequencing. So originally it was based on standard method, like old school methods like geoelectrophoresis, in vitro reactions and so on. But with arrival of next generation sequencing methods, and especially single cell methods, this area was actually really boosted. I'm sorry, can you hear me well? My voice is a little bit, okay, that's great. So majority of methods for chromatin profiling could be summarized in that way. You isolate nuclei, I'm sorry. Yeah, I apologize. Majority of chromatin profiling methods could be summarized in the way you treat isolated chromatin or permeabilized nuclei with enzymes which could either cut or modify DNA. So that could be DNase 1, mononuclease, transposase, as well as different DNA modifications enzymes like DNA methylases. And basically, as much chromatin is, as much accessible chromatin is, as more cuts or modifications you have in this region. So basically, after this type of treatment, you isolate DNA, make library from either fragments or you fragment it separately and make library, and then do high throughput sequencing and assess chromatin accessibility. Like here on this profile, if we have long access coordinates, you could see some regions are highly accessible with open chromatin, and other regions are not accessible. So this is the base of DNase sequencing, ATAC-seq, and so on. And there are multiple of these methods, and recently the biggest popularity obtained ATAC-seq because of its simplicity. So I will remind you, this method is called assay of chromatin accessible to transposase 5, and basically it uses transposase 5 to cut DNA. So transposase 5 is actually used for a number of methods, and basically by preloading hyperactive transposase with pieces of DNA that serve as PCR and sequencing adapters, you could very efficiently both fragment DNA and insert adapters. And this is used in a number of genomic techniques, but for ATAC-seq, basically you incubate cells or chromatin with adapter-loaded transposase, and you got insertion proportional to the chromatin accessibility. So this method was developed by Jason Blanrostro at 2013, and since that it uses in a number of areas. Besides ATAC-seq, there are historically and currently there are a number of other methods developed for probing chromatin, and some of them give different information. So while ATAC-seq and as well as earlier developed DNase-seq mostly allowed to obtain fragments mostly from accessible regions, for instance, micrococcal nuclease is other enzyme which is used for it, and it's so active that you basically don't get any fragments from accessible from open regions, and whatever fragments are you get is basically footprints of nucleosomes. And therefore scientists sometimes combine these methods to get better understanding of actual chromatin structure. From methods like ATAC-seq and DNA-seq, you could get actually a number of information, different kind of information. So first of all is accessible regions of chromatin. Like again, you could look on where you have peaks of accessibility along the different genomic coordinates. Like usually you get as accessible regions, promoters, enhancers, and actively expressed genes overall. Also, besides that, one could use use this information for inferring nucleosome positioning, because again, regions which are covered by nucleosomes are usually less accessible, and once you look on the size distribution of fragments coming from methods like DNA-seq and ATAC, you could see distribution, this kind of phasing of lenses with increased frequency of lens, which correspond to mono, D, and so on, and trinucleosomes. And by analyzing the fragments coverage, the coverage of region with particular fragments of ATAC-seq or DNA-seq, you could understand is this region actually covered by nucleosomes, or it's more compacted, and so on. So in this way, you could infer nucleosome coverage profiling from ATAC-seq. And also, there are a number of methods to infer transcription factor binding sites. This is actually more tricky method, which work for some transcription factors, but not all. So basically, by a local decrease in accessibility, which is usually pretty tiny, and with by assessing coincidence of this decrease with transcription factor motifs, one could predict with more or less good probability binding site of particular transcription factors. This is so-called footprint analysis, and it's pretty good for some transcription factors like CTCF, but for transcription factors with less defined motifs and transcription factors which are not very strongly binding to chromatin, this could be more challenging. So overall, from open regions discovered by ATAC-seq and other chromatin profiling methods, you could infer enrichment of motifs of transcription factors. And this is important to understand the actual regulatory pathways of that govern gene expression, and basically to understand what transcription factor mediates opening of this region and activation of the surrounding genes. And this is usually done by motif analysis tools, either customly developed or the most popular, well used are Homer and MIM suite. So usually people compare enrichment of motifs within regulatory regions or discovered differentially open regions to all the rest of regions across the genome. And one could do de novo motif discovery, like figure out what motifs are which k-mers or which motifs are someone is drawn on the screen. This is weird. Okay, which motifs are enriched, and also to figure out what known motifs previously discovered transcription factors are enriched in particular region. So this method within the last 5 years was applied on single cell level. And basically, I will briefly mention why we care about single cells. So pretty often we deal not with uniform cells grown on plates, but with pieces of tissue, like a tumor or healthy tissue, or developing embryos where we have population of different cells. And those cells have different gene expression patterns and different chromatin profile. And if we analyze them all together in bulk, we will get some average profile, which would miss some more less sparse cell populations. But however, if we will be able to separate cells on different populations, we will be able to access actual state of each cell population. And in that way, for instance, here we could create aggregate groups of particular cell types and analyze them separately. So talking about Ataxic, first attempt to apply it for single cell level was done also by Jason Van Rostra. And it was pretty much classical single cell approach where they used microfluidics device to separate cells and cell lysis and loading of the transposase loaded by adapters was also done at the microfluidics device. And then after treatment with transposase, cells were lysed, library was prepared, and they did sequencing. So because unlike the RNA, DNA is present in cells usually in 2 copies with some exceptions. Basically, what you get from each single cell is basically 2 copies of different fragments. So it's not like you could you have an entire population of RNAs coming from the particular genes. So in that way, signal is pretty sparse. So technically, besides PCR duplicates from each cell, you get basically 2 DNAs with different cut sites. And in that way, it's pretty difficult to analyze for one single cell. However, you could analyze fragments and put them into and separate them into different populations, like with aggregate analysis. And already these populations you could profile for different open regions. Since that approach, there are a number of other approaches to implement Ataxic. And actually, they were also commercialized, for instance, by 10x Genomics, which developed the commercial Ataxic platform, also based on the droplets, on the barcoding and chromatin profiling in droplets. So this could be applied for either separate cells or single nuclei isolated from cells. And barcodes are attached to magnetic beads, and basically these beads could be loaded on the to the droplets with single cells. There are different other approaches for perform Ataxic. One of them, which is promising for especially combining single cell Ataxic with other single cell approaches, and actually is not requiring any special separate equipment like microfluidics. It's called SCI, which is for single cell combinatorial indexing approach. This approach was developed a lot in Shindura lab and some other labs. So basically, principle is very simple. You need at least two sets of barcodes that could be introduced by transposition and by PCR, and you need 296 or 384 well plates, at least two. And principle actually here it is the same for both Sci attack or Sci RNAseq, and this lab actually developed different methods based on this approach. So basically, you pipette the suspension of cells into 96 or other multi-well plate, so that each well would have a particular small number of cells, like around 20 cells. And each well has specific barcodes, which is introduced, like in case of Ataxic, by transposition. And then after first barcoding, you again mix cells together and again pipette to other 96 well plate with also other barcode combination. Very often, this barcode introduction is done by different steps of the protocol. Like the first step could be attack, and second, either second transposition or already one of PCR steps. And in the end, you combine all the cells or DNA from all the cells, and you have barcode combinations from first and second plate, and with special adjust cell number for each well, you could get conditions when each cell have its unique combination of barcodes. So basically no microfluidics, no other equipment is needed. You just need to do 2 or 3 rounds of barcoding. And basically, authors of this paper from Shindora lab, they were able to reach good enough separation of single cells this way, so that, for instance, once they mix population of mouse and human cells, they were able pretty decently separate mouse and humans, like you could see from this plot. By the way, could you see my mouse? Okay, awesome. So this Sci method was used pretty actively for later combinations of Ataxic with other methods, because you could also combine sequential introduction of barcodes with a sequential introduction of barcodes to different molecules like RNA and chromatin. So we talk a little bit about chromatin accessibilities, but I have to remind you that there are other chromatin marks, or other levels of regulation of chromatin, and this would be first of all histone modifications and proteins associated with chromatin. So you probably know this picture pretty well. Histones are used extensively to be targets of secondary covalent modifications. They have pretty long tails, where different histone modifying enzymes attach different modifications, which is acetylation, methylation, phosphorylation, and many others less known. So typically, histone modifications and other epigenetic modifications have this type of life cycle. There are complexes which introduce the mark, so-called writers, complexes which remove the mark, and also protein complexes and particular regulators which recognize the mark and change region, either by recruiting chromatin remodeling complexes or transcription factors, POL2, and so on. So more or less for each histone marks, there are also their own reader-writer erasers, like for very well characterized lysinase acetylation in histones, there are enzymes of so-called histone acetylases, which introduce acetylation, and histone deacetylases, which remove acetylation. And basically, they are differentially recruited to different genomic regions, and they are extensively used for regulating gene expression. And altogether, histone marks are pretty often recruited on histone modifiers are pretty often recruited to particular regions by either transcription factors or small non-coding RNAs. I will briefly talk about transcription factors. These are usually DNA binding proteins which recognize particular binding sites. Usually, this is particular motifs within DNA, and after they bind, they on its own recruit different chromatin activators and repressors. And sometimes binding of a transcription factor to inactive chromatin could trigger chromatin opening and activation. And in this case, transcription factor is called pioneering transcription factor. By analyzing transcription factor binding, one could infer a signaling pathway which governs changes in gene expression and figure out the regulatory pathways of which activates particular genes in certain process. Therefore, there are a number of methods that analyze binding of transcription factors, and as well as histone modifications and binding of other proteins. So well known and historically pretty and the method which historically was used pretty long ago is so-called chromatin immunoprecipitation. It was used even before NGS, just by PCR, and with NGS we got so-called ChIP-seq, chromatin immunoprecipitation combined with whole genome sequencing. So very briefly, cells nuclei are cross-linked. Transcription factor is bound by a specific antibody, and basically you immunoprecipitate transcription factor in complex with a fragment of DNA which it binds, and then DNA is isolated and sequenced, and you understand after sequencing, you could see which regions are enriched, and that would be the transcription factor binding sites. So ChIP-seq, while it was used pretty often still, and it's still very popular, it has its own bottlenecks, and especially for single cell approaches. So overall, it's pretty long and sensitive protocol, and quite often for particular proteins or for particular biological model, it requires some optimizations. And one needs very good antibodies. So basically everything depends on how good antibodies are, and those antibodies should recognize proteins after formaldehyde cross-linking. So this is so-called ChIP-grade antibodies, and pretty much very often antibodies are not specific enough. They recognize other proteins and give background, and one needs a lot of controls. So this procedure is pretty challenging for single cell adaptation. Even so, some groups adapted it for single cell methods. However, recently some alternatives for ChIP-seq arise, and you probably heard of cut and run and cut and tag. So basically, in this case, you don't do immunoprecipitation. One just treat cells or nuclei with antibody covalently bound or non-covalently bound to either microconcave nuclease or transposase 5. And this enzyme, together with antibodies, is bound to regions which were protein seeds on chromatin, and in that way it introduces cuts around target protein binding site. So with micrococcal nuclease, later on you need to make library of these fragments of DNA, and with TN5, TN5 itself is introducing PCR and sequencing adapters, so you need just to amplify these fragments and use for sequencing. So this is a comparative scheme of ChIP-seq, cut and run and cut and tag. Yeah, basically cut and run and cut and tag that differ with enzymes that are used, and overall these protocols could save a lot of time and are so much simpler than ChIP-seq for implementing, and the appearance stimulates whole genome protein binding sites profiling, because they're so much easier to implement on single cell. For instance, single cell cut and tag method combined with combinatorial barcoding, like used with multi-wall plate. So it allows to screen for transcription factor and histone modifications binding sites across across the cell population, and this could be also combined with other approaches and to profile combinatorially RNA expression and protein association. Just a moment. Okay. So very promising direction is to have joint profiling from the same cells in the population of chromatin accessibility, RNA expression, and other parameters. Because this allows to understand actually which mechanisms underlies different gene expression changes in different cells. Arrival of this method is based on progress in single cell profiling mechanisms, so like the methods like Psi, and yeah, they basically came in last 3-4 years, not earlier. So for instance, the PsyCar, which stands for chromatin and RNA profiling from Shinduro, is based on the combinatorial barcoding with Psi method, and basically you use oligo-DT primer for reverse transcribing RNA, and also at the same time one uses TN5 transposase for profiling chromatin accessibility, and altogether you could you get together RNAseq and Siteseek libraries that you could trace by barcodes, and you get information from the same cells with both RNAseq and of both RNA expression and attack. There are a number of other methods like pair-seq and so on that also do the same. So this approach is still developing, and papers appear about combinatorial profiling of different marks. And I have to mention here that some of these approaches also try to profile DNA methylation together with RNA expression and chromatin. I didn't mention about DNA methylation before, so usually on its own, it is usually profiled by bisulfite sequencing. So DNA methylation is covalent methyl group attached by DNA methyltransferases to cytosine. And in higher eukaryotes, usually you get methylation marks mostly introduced to repetitive elements in heterochromatins, and also in CPGs within CPG islands. And especially CPG islands are important for regulation of expression of nearby genes. And usually, mostly people are interested in profiling methylation in CPG islands. Classical method for profiling DNA methylation is so-called bisulfite sequencing. With bisulfite conversion, unmethylated cytosine is converted to uracil, and methylated cytosine is protected. So after amplifying bisulfite converted DNA fragment, you see unmethylated cytosine common as a Ts and methylated cytosine as C. And this method was used in combination with NGS, and more recently it was also adapted for single cell method, so-called single cell bisulfite sequencing. So again, like you separate cells with microfluidics or whatever in single cell, and DNA from single cells is converted with bisulfite, and then you have specific adapters attached by which you could separate individual cells, and yeah, you do library and sequence and analyze. So recently, this method was combined, for instance, with RNAseq. So this is especially valuable to understand how exactly in each cell methylation methylation influence on RNA expression. And so-called, for instance, SCMT-seq or SCMNT-seq. So you isolate nuclei, separate cells on the single cell population, isolate nuclei and cytoplasm, and from cytoplasm one prepare RNA library, usually by SMART-seq approach, and from nuclei one does bisulfite sequencing, and yeah, combining these methods, one could figure out how methylation of individual CPGs affect RNA. More recently, attempt to combine methylation with chromatin accessibility was done. So it was based not on chromatin fragmentation, but on modification of chromatin with other methylase, which is in contrast to physiological CPG methylation, was introduced in GPCs. It is so-called MCVIPI methylase, and basically by treating nuclei with this methylase and followed by DNA isolation, bisulfite conversion and sequencing, one could infer at the same time CPG methylation within CPGs as physiological methylation, and GPC as a proxy for chromatin accessibility. So of course there are sites where you have a number of GC coming together, but authors conclude that this is minority, and altogether this method is pretty useful to see how methylation affects chromatin accessibility. So here comes promising part for this method. So while bisulfite conversion was used pretty often before, it still has biases because it's like chemical conversion, and if you, for instance, if you change little bit conditions, your profile of methylated cytosines could differ a lot. So this method is biased, is prone to biases. So here comes a new method based on the Oxford Nanopore platform. This is so-called direct sequencing of DNA. So basically it's based on the passing DNA through the pore with assistance of motor protein, and as DNA passes through the pore, changes of electric current occurs on the membrane, and from these changes of electric current, you could see, you could infer actual sequence of DNA, and not only you could infer any modifications which take place on this DNA strand. And this way was actually used successfully for inferring CpG methylation, but not only, so you could also use it for other type of methylation. So one could use so-called nanopolish tool for this inference, and authors of recent paper successfully adjusted it for both GPC and CpG detection, and in that way one could detect a profile of both nucleosomes and chromatin accessibility and DNA methylation at the same molecule of DNA. I have to mention overall the promise of this long read approach. So this is both Oxford Nanopore that I mentioned and PacBio. So this approach, long reads, allow us to use to profile modifications wherever in long fragment of DNA, which could be multiple kilobases or even up to megabase, and this gives us such a concept as a single molecule sequencing. So instead of multiple short reads that you get from Illumina, you got one long read, and you could trace all different modifications or all different chromatin state across all this long molecule. And technically for DNA, this approach could be used as some kind of alternative to single cell approach, especially for tissues which are hard to lyse or where which you cannot separate on single cells by some reason. So basically, you cannot use it for RNA because RNA is present in multiple copies per cell, but DNA is usually like you have 2 copies of DNA, and assessing profiling the chromatin state or methylation across these long DNA reads, you could then separate the patterns in subpopulations and use these subpopulations in the same way as you use for single cell sequencing. And in that way, this approach is also promising and could be used instead of single cell sequencing. I think I have to wrap up my lecture. So I have to mention that advance of all these methods give rise to big projects like Human Cell Atlas, which aim to get a profile of all different cell types within human body. in normal conditions and disease. And also there are big projects for mice and other organisms. However, there are still limitations in single cell profile methods. So of course, it is still yet low coverage for most of the methods, especially methods based on DNA rather than on RNA profiling. A lot of reads drop out, so you don't have complete coverage for each cell. And yeah, basically to do analysis with more or less robust significance, you need data from population of cells out of the entire population, this is rather than accessing what's going on in each individual cell. So if you have one cell per entire population, it's more difficult to get data from it. And this method is also difficult to implement to some type of tissues which are hard to lyse and organisms like plant and fungi. So here is the space for single molecule long-read sequencing for these organisms. So with this, I guess my time is ending. So I would like to thank you for giving me opportunity to talk to you, and your questions are welcome.
Yeah, thank you, Yulia, for such extremely informative presentation today. So we have some very nice questions in our chat. And
I think we...
Yeah, please go ahead.
Before we forward to the Q&A session, we have something also to ask our participants, isn't it? We would really kindly ask our participants to evaluate the meeting today.
Oh, sure.
Before we go to the Q&A session. And I would like to share also the screen with the QR code for the evaluation. So Julia, if you could unshare your screen, please, I could do it. Thank you very much.
Okay.
So guys, we are very, very thankful to you also from the previous session that how you evaluated our day one lectures. We saw a lot of response from your sides with positive and negative feedback. And we are really very glad that your evaluation is so good. And we have a lot of ideas how could we do our lectures even better. So at this time, I would also be very glad if you evaluate our webinar today with part 1 and part 2. You can access the evaluation form with the QR code here. And feel free to write also your suggestions, your negative and also positive feedback, and of course, your overall impression from the webinar today. It means a lot for us. We are reading every evaluation through, and then we are happy to improve our lectures for the future. So thank you very much once more for your evaluations and for your participation today. We will also duplicate the link into the Zoom chat and into the Slack channel so you can access it without the QR code.
Okay, so should I use the rest of time to answer questions?
Yep.
Vladislav, shall you moderate the Q&A session, please?
Of course, I could do it. So we have some questions in the Slack channel gathered for you, Julia, and I would just go through them and we can discuss them together. So One moment, please. So all the questions are also gathered in the Slack channel under one post. So for you, it's of course even better than to track the questions. I mean, the participants in that case.
And I just would like to mention that it's easier for us to have all of your questions in Slack, because after the Zoom, after our Zoom meeting is ended, we simply lost your questions. And that is why all of your feedback is very welcome. But please, if you would like to receive an answer to your question, it's more like the higher chances that we will be able to reply in Slack. So please, dear participants, post it there and we will be glad to reply them directly. So yeah, please go ahead.
So question number 1, if in ploidity disorders, epigenetic factors play a crucial or significant role, could we use this technique to discover proportions of genetically abnormal sex cells for a given time and space in a patient or in population?
So can you use these techniques for discover the change in DNA sequence? So I guess there are techniques,
especially in the ploidity disorders.
Yeah, there are ways to infer the ploidity information from Ataxic. So you could also use it for the single cell, like just single cell DNA-seq as well. So basically you infer it from the coverage information, and especially like if you have aneuploidy, you could infer. I guess it's better to use not Ataxic, because here you could also, you have also information from like factor of accessibility of different regions. But I would use like just single cell genomic DNA sequencing, and you could access the coverage in different regions and infer the ploidity, like aneuploidity for different regions.
But I mean, in that case, it's of course very interesting that you're saying that the DNA sequencing would be better, but don't you think, for example, that you can't detect the abnormal cells so good with the DNA sequencing as if you would be doing it with the Ataxic?
Well, like primary sequence, yes, for ploidity, like I would be careful because like if we're talking about the Ataxic, single cell Ataxic, here you also have a factor of chromatin accessibility. So for instance, you cannot, you would have less coverage definitely in heterochromatic regions, but yeah, you could, I think one maybe could infer the ploidity, like aneuploidity, and one definitely could get information for like genetic variants like SNPs and so on. But yeah, actually Ataxic based method was recently successfully used for profiling insertion sites of lentiviral vectors in CAR-T technology, like to access where the lentivirus landed within the like in the CAR, and to access the chromatin activity within surrounding region and lentivirus itself. So this, yeah, modifications of Ataxic could be definitely used. One need probably to adjust it to a particular approach, but yeah.
Okay, so the next question refers to one slide where you showed the 96 well plate. And the question is 96 well plate coated with specific transposases?
So usually people use same transposase TN5. One could, if one has access to unloaded TN5, or like one could isolate itself in the lab, TN5, one could produce like to load it with different adapters, with different barcodes or UAPI. So this would be one way, like to put transposase with different adapters in each way, in each well. Other way would be to use the same transposase, but then use different UMI primers for amplification. So this is also possible.
So basically, the both approaches are right. You may coat with transposase the well plate, but you can also use it in just normal master mix.
Like usually to have transposase with different indexes, with different adapters, with different indexes, you need to have access to isolated TN5s that you load it yourself. So this is technically more difficult. And not everyone could access it. So like if you work on company, you should use transposase only within Nextera kit because of the proprietary reasons. So, but if you have access to, if you isolate yourself TN5, you could preload it with different adapters and use like that. Otherwise, if like it could be the same transposase with the same adapters, but then you could use like later steps like PCR preamplification with different adapters.
Okay, good, thank you. The next question would be a little bit broader one. What are the negatives of the ATAC-seq?
Negatives. So what are the downsides? Like there are definitely biases. So first of all, if you use a single cell approach, it's pretty easy to drop out some regions of the genome just because you have just to DNA from each cell, and it's very easy to lose the material. Second, transposase itself has some bias, which is probably not very big, but still accessing the chromatin patterns and like enrichment of motifs. One need to keep in mind that transposase have like some slightly preference to particular sequences. So sometimes this adjustment is for transposase preferences used in analysis methods for like inferring motifs from accessibility profiles and so on. Yes, there are definitely some biases. Yeah, I could think and more detailed answer in the Slack.
Good. The next question is: do we need the TN5 to do the bisulfite-seq?
No, bisulfite sequencing it is done with isolated DNA, and basically it is based on the bisulfite conversion with bisulfite treatment. One could maybe somehow combine TN5, like basically TN5 is used for rapid genomic DNA sequencing library construction. Like one could do it, one could then briefly treat with TN5, insert adapters, and use it for PCR, and this could be done instead of like old school, like fragmentation, ligation, and so on. So this would be definitely easier, but it's not technically, it's not necessary.
Cool. Okay, so we're coming to the next question, if it is okay for you, because we exceed.
Yes. Okay. Now I'm coming the last one, like I have time. Yeah.
Okay, good. Then the next question would be, what about the methylation of adenine? Is it under less investigation compared to cytosine?
So if we're talking about DNA of higher eukaryotes, methylation of adenine is not found in this organism. If we're talking about bacteria, this is different question. But again, like a lot of promise here comes with nanopore sequencing, because you could infer modifications of DNA and actually RNA as well from this profile of DNA passing through membrane to affecting current. And this, like, if I would start studying methylation in bacteria, methylation of adenine, this is something that I would be using.
Okay, so that means that when we are talking in the human context, then the CpG methylation is much more important as other methylation types.
There is no adenine methylation. There are a lot of conversations in the field that like there is non-CpG methylation, and some people don't believe it, some believe. But basically CpG is like everyone, like at least acceptable in the field is like CpG is the only methylation. Like some people occasionally publish papers that they find something else, but yeah.
Okay, so there is a little bit room for conspiracy there, as I understand.
Yeah.
Okay, so the next one is it possible to do a single cell mitochondria ATAC-seq in order to identify mitochondrial DNA variants in a cell?
Well, this is a funny question, because mitochondrial contamination, this is a big problem sometimes when you have ATAC-seq data, because you got mitochondrial DNA and you have to subtract, and sometimes mitochondrial reads are predominant reads that you have in your sample, and this is very unknown. It's the same as for RNA-seq ribosomal RNA reads. But definitely, I guess it could be used. But what's going on in mitochondria is much less studied, like how much mitochondrial DNA is compacted and what is there overall. Like definitely there is a field of further study, but yeah, even within standard attack you get mitochondrial reads, and if you're curious enough you could try to play with them. Yeah.
Interesting. Okay, so that means that you are accessing normally the mitochondrial reads also with the single cell or just normal attack-seq, but it's only the matter of mapping in that case.
I guess what happened, like ideally you want just to have like isolated nuclei and permeabilize them and treat with transposase and don't have mitochondria. But basically, I guess once you lyse the cells, once you pellet nuclei, you could still get some leftover of mitochondria and get it. And more recent adaptation of ATAC-seq, they even don't separate nuclei, they use entire cells, and then like you have plenty of mitochondria, and yeah, you get also signal from mitochondria.
Okay, so the next question would be in doublet, because these two questions are really very related. The first one is, are there any commercially available products that combine the single cell RNA-seq and single cell ATAC-seq in the same cell?
So let me look closer and reply on Slack. So there is definitely one which combines methylation and RNA from Illumina, but yeah,
but ATAC-seq and RNA-seq, as I know, for example, from the 10x, you can't do it simultaneously in.
Yeah, they recently announced this multiome kit, like it's happened in September last year. So actually, it's already commercial available kit. So
okay,
yeah.
But do they allow to do this ATAC-seq and RNA-seq in this same cell?
Yes, in the same cell.
Ah, cool. Okay, good.
Yeah, so that's the wonder. So like one of these, as Julia mentioned, one of these crucial points is quite often you have a lot of dropouts. And that is why by combining these two methods, you can overlap and even impute your data of gene expression in much more comprehensive way. So that is quite promising tool.
Okay.
Yeah, we could maybe like chat more for this in the Slack about the actual commercial products, because yeah, I'm not using myself commercial single cell RNA ATAC-seq, but yeah, would be great to discuss this further.
So the next question is related to the previous ones about the available tools for the simultaneous sequencing. The question is a little bit specific, so let's start. From your point of view, which method is better to study neurodevelopmental disorders using multi-omic single-cell resolution? So droplet-based or combinatorial indexing ones? For example, 10x chromium single-cell multiome attack plus gene expression droplet-based method, or recently developed shared-site profiling of RNA and chromatin?
So somehow I have my own preference to the combinatorial method, because yeah, you could get data from a lot of cells, and you don't need either special equipment or like buy product or like get the service from 10x Genomics or so. And you can get decent data, but maybe like for some labs, like they want to rely on actually well-established commercial kit or so, and that like it's especially would be important like if this will come to the clinical like for clinical procedures, which is not the case now. But yeah, I myself I would use combinatorial, but yeah, it also really depends on the tissue and so on. So like yeah, and exactly what you want to profile, but yeah.
That's really a very interesting question, because it was highlighted the field of the application, and maybe the good question would be also, what is your point of view to the the best field of the application in single-cell ATAC-seq. What field would prefer the most from this method? For example, I know that in immunology, some of the T cells may differ in their epigenomic profiles, and it could be really a very good marker to distinguish between some of the subpopulations of the T cells. But what is your point of view? What is the best field for the application of this technology?
So of course, like it would be probably any field which studies heterogeneous cell population and like cell lineages tracing. So if we are talking about disease, so cancer, of course, like because you have heterogeneity within tumor, and you have it's nice to trace like cells which start acquiring resistance or start getting secondary mutations, and it's not necessary to be mutations in DNA sequences. It could be like changes in epigenetic state which you could detect. So this is about disease, but talking about like general biology, of course, it would be development of like different stages of development of embryo, like early stages, development of brain, also develop like blood differentiation, because again, like you have lineages of different blood cells, and it could be like bone marrow profiling or like blood profiling and so on. So yeah. So hematology.
Because of the transcriptional factor sites, for example, that can be revealed by this technique, or
yeah.
Okay.
So yeah, there are special adaptations for motif analysis for single cell methods that I didn't talk here, but I could post in the Slack for some papers or some GitHub.
Because I think that many people are asking themselves, why should they do the single cell ATAC-seq if they can do the single cell RNA-seq? Why should they access the DNA epigenetic information if they can access the product of this interaction of the DNA transcription and epigenetics on top?
And the question is absolutely legit. So, so who wants to assess ATAC versus RNA? So, like some people, like who are working in academia, like they study mechanisms of regulation, and then combining ATAC with RNA-seq or like with other methods. So this is basically for the basic knowledge. This is one. Second, in clinical applications. So for mammalian cells, for majority of mammalian cells, I guess ATAC-seq might be still easier than to do RNA-seq, because you get less signal from each cell, but like chromatin is like DNA is more stable than RNA, and basically ATAC-seq procedure is very simple. Like you could just lyse cells or even not lyse cells, incubate with transposase, and then it already introduces cuts and adapter insertion, and then you just do PCR and proceed. So if you want some simple procedure which would be still robust readout, bulk ATAC-seq and even single cell ATAC-seq might be still okay. So this could be adapted for particular like assay in clinic and so on.
So you mean the accessibility and the stability of the techniques is one of these major benefits?
So for single cell might be different. Like for bulk ATAC, definitely bulk ATAC is easier than bulk RNA sequencing. Single cell, because again, because DNA is present in much smaller copy numbers, maybe it's comparable, maybe it could be easier. So it could, in some situations, it might be easier as a assay approach.
So, but could you maybe like highlight one of the or two things that ATAC-seq can give you, but the RNA-seq not? Yeah, like the information outcome that you can gain only with a single cell ATAC-seq, but you cannot reach it out with the messenger RNA and transcriptome sequencing.
It could be, for instance, activity of regulatory regions like enhancers. This is usually non-coding regions, but they influence expression of nearby genes, and usually they are bound by a number of transcription factors, and they could be looped together with the promoters. So like they could be on distance, but in close proximity. And usually on Ataxic, they are also, if they are active, they are they're accessible on Ataxic. So yeah, this is one thing. Also, like you could look on the activity of promoters yeah.
So this is like the main benefit, that you are getting something more spatial in that case, that you are getting a position where your activity is happening on the DNA strand.
I would say so. Like in general, methods for chromatin profiling, they are more used by basic scientists who are studying this chromatin and epigenetic mechanisms. However, talking about how it could be used, let's say, in clinic or clinically oriented studies. So, mechanisms of diseases like some types of cancer and so on. So they are epigenetic. That's why, like, there are a lot of compounds which are like inhibitors of histone deacetylases, inhibitors of DNA methylase, and so on. So this is like some type of anti-cancer therapy. And for this type of cancers, then like you are interested in like what's going on epigenetic level, and Ataxic, I guess it's a pretty good approximation for whatever goes on about chromatin state. Of course, it's for a more complete picture, it's more interesting to combine it with histone methylation, histone modifications, and so on. But Ataxic, probably it's kind of it's a easy proxy for understanding what's going on.
Yeah, very interesting. Yeah, thank you. I think when we are speaking about the disease, we can also turn back to the very first question that we talked about the aneuploidic diseases, because Alexander has something to say also about this.
Okay.
So if you could please unmute Alexander. Yes,
Hi,
we hear you.
Greetings to the participants. Thanks for the lecture, Julia. I was able to participate only on the second half, unfortunately, but I liked it a lot. So it just happened that I really enjoyed the first question regarding copy number variants in the sex cells, because I have worked in the reproductive medical center for 3 years, and I was always thinking, like, why not use a next generation sequencing just to, like sequence any suspicious embryo or whatever. So here we actually face a few small problems. Like if we talk about the sex cell, we can start with, I don't know, oocyte, for example, and the question is material. From the oocyte, we can only retrieve the polar body, which is haploid. So we'll receive the half, let's say half of the person genome material. And if we even receive the feedback, there is no aneuploidy, like normal copy number of the chromosomes, we will still not receive any information about the future embryo, which may be aneuploid after fertilization. So usually people do this only in the case of legal regulations. For example, in Germany, you just cannot make the biopsy of embryo, and people do the biopsy of the polar body. Another question is embryos, because there we can actually make a biopsy and retrieve either blastomeres or trophectoderm. So usually NGS here works pretty well when we're talking about the normal copy variants or normal embryos. So when we take aneuploid or embryos with abnormal chromosome number biopsy, we usually work with non-equally distributed mosaicism. So we can have quite severe mosaicism, which is quite difficult to detect with NGS, but quite easy to detect with genome hybridization methods. So that's why people usually prefer using arrayed, what's the name, ACGH, arrayed comparative genomic hybridization, I believe that's the name, because it's much more sensitive for very bad mosaicism, where some cells are aneuploid, some cells are euploid. So for this reason, people not usually use NGS for such occasions. And if we speak about sperm cells, so male sex cells, it's quite interesting for scientific purposes, but it's mostly used for medical purposes, because you never know if you fertilize with aneuploid cell or normal cell, even if you diagnose, for example, there is half of the cells with aneuploidy. So it's just impossible to check. There are some non-genetic tests to review this. However, they're not exactly informative or sensitive enough. So here we don't have a good answer. So just to sum up, NGS is super promising for that, but unfortunately not yet, not unless we know how to manage with really bad mosaicism in the analysis, or maybe to modify somehow our protocols for that. Yeah, that's all for me. Thanks.
We are talking about the NGS, if I may, or did you want to say something, Julia? Sorry.
Oh, I think it's like it's quite a nice discussion. I like it. So thank you, Alexander, for your input.
Okay, so when we are speaking about the NGS, I have one question of our participants left, namely, which type of sequencing is the best option to differentiate the stem cell populations in a tumor, according to your opinion?
So till now, Illumina basically is the dominant on the market, and I think they are probably still the easiest to use because there are a lot of services where you can send your samples. If we are talking about like Illumina versus Ion Torrent versus Nanopore versus wherever. So Illumina provides like a lot of different kits and so on. So Oxford Nanopore, like I guess they are starting to take market, but still this technique is still a lot of under development. A lot of approaches are still like community based and so on, but they have a lot of their own promise. If we are not talking about like accessibility of technical support and so on, I think Illumina in the end might be comparable to Oxford Nanopore, and Oxford Nanopore is good. Like you could profile long read, like with all its pattern of like methylation and single nucleotide change and so on, and you could have it like one long read coming from this one cell. And for Illumina, you need still to infer from which cell comes all of the small reads. And yeah, so I would say they would be maybe at some point comparable, or maybe Oxford Nanopore would have advantage at some point, but let's see.
Cool. Okay, thank you for your opinion, and thank you for your answers to all of the questions. I know that we overexceeded today the time, but still it was really very interesting to hear your answers and your opinion to the questions. I think we even answered all the questions that we gathered so far in Zoom and also in Slack. So if you have any other questions, guys, just put them in Slack, and afterwards we can also answer it after the webinar today. So I think it would be a nice point to make a cut today and to end the meeting for us. So from my side, thank you all the participants that you came for the meeting and that you participate in our course. We value it really a lot, and that's why I'm happy to see you also in the next courses. And I will give the microphone to Julia.
Yeah, thank you so much for inviting me and for this nice discussion. And I was very excited to talk to all of you. Yeah, and please ask your questions. So we've decided to continue some discussion about commercial kits for simultaneous Ataxic and RNAseq. We sort of continuing it on Slack, and we can do it. And please answer further questions, and I will be happy to talk to you.